Good afternoon. Thanks so much for joining us. I'm Robynne Chutkan, an integrative gastroenterologist on faculty at MedStar Georgetown Hospital and based locally in Washington, D.C. The pleasure of having some luminaries and some friends here on the panel. To my left, I have Mark Hyman, who is the head of strategy and innovation at the Cleveland Clinic Institute for Functional Medicine. We have Rob Knight, who is one of the foremost microbiome researchers in the world and on faculty at UCSD. David Perlmutter, who is the Dean of Medical Affairs at WashU, and also pediatrician, gastroenterologist, and Karen Sfanos, who's a researcher at Johns Hopkins in oncology and prostate cancer specifically, but who spans oncology, urology, and pathology. So I'd like to start off by asking all the good people in the room to raise their left hand and all the bad people in the room to raise their right hand. So good people, raise your left hand. Bad people, raise your right hand, right? So this is a little bit of the challenge we face in trying to figure out the microbes. We know that of the trillions of microbes that inhabit our body, and you all know the statistics, we're really more microbe than we are human if we look at the genes, microbial genes versus human genes. But trying to figure out who's friend and who's foe. We know that microbes can do some bad things. They can cause cholera and tuberculosis, et cetera. But 99% or more, I don't know if you'd agree with that number, Rob, of our microbes are probably working symbiotically with us. But again, trying to figure out, industry has sort of distilled it into this good versus bad, and it's far more complex. The two questions I really want to try and get to today with our really distinguished panel is, number one, what should we be eating? And number two, can we eat in a way to actually prevent disease, to prevent developing disease, to prevent disease progression? And if so, what does that diet look like? So Mark, you've written tons of books, New York Times bestselling author. Your last one talked about fat, actually, and that food, fat was actually, yes, food.
I've read so many, it's like you can't keep track.
Focused on fat. But I think there's no such thing as an ideal diet, right? But how close can you come in your estimation?
Well, what's really striking with the microbiome is that, you're not just feeding yourself, you're feeding a whole inner garden. And what you eat determines the bugs that grow or don't grow. And that determines the quality of your health. And so it's something that we're just recognizing now. It's not as simple as just taking probiotics or even prebiotics. It's the food components, the polyphenols, the flavonoids, the various phytochemicals, the fibers, all regulate what grows and what doesn't grow. So the fact is that most of our good bacteria that produce all the metabolites that keep us healthy and produce vitamins and regulate our hormones and regulate our metabolism are controlled by eating a lot of the plant-rich foods. And I wouldn't call it plant-based, I would call it plant-rich, which really includes lots of colorful plant compounds that are in fruits and vegetables, and lots of high-fiber foods that also provide some of the prebiotics. And it's striking when you look at the individual response of different microbes. And I've had personal experience, I can share a little quick story, but I had developed colitis a couple of years ago, after a number of stresses and insults, and I've had a very low level of this one particular microbe called Akkermansia that is involved in protecting the biofilm and regulating immune function, and has been linked to autoimmune diseases. And I'm like, wow, how do I get more of that? And it turned out from the literature that they respond to certain polyphenols like green tea, cranberry, pomegranate. So I created a cocktail and added some prebiotics and probiotics and a few other things. And within 3 weeks, it was completely cured. And
his favorite microbe, Akkermansia.
And I rechecked it and it went up. And I've been using this with patients. So that's just one example. And that that could be multiplied literally probably millions of times in different nuanced ways, in ways that Rob and I were talking about before, because so many of these microbes have unique needs and need unique fertilizers.
Karen, you work specifically on the effect of the microbiome on the food, and then how that down the road interacts to cause or prevent prostate cancer. Can you talk a little bit about that interaction, the food microbiome interaction?
I will talk about that, but first I'm over here shaking my head at Akkermansia because there's a story there in cancer
as
well with
immunotherapy. Yeah.
Mm-hmm. And a paper came out to say that, and the Akkermansia is interesting because some people seem to have it and some people don't. It's like a yes or a no, and the people that do have it, you can increase it. We don't know really what to do with the people who who don't. But with immunotherapy, there was a paper that came out that said that those that do, that have a lot of it, tend to respond to immunotherapy for their cancer better than those that don't. We don't know the mechanism, but it's a very interesting story. And ellagic acid for increasing it.
That's green tea.
Right, grapes, pomegranate. On the flip side of that, one of the things that we study in the lab in the context of prostate cancer is what microbes can do with food that actually aren't so great in terms of cancer risk. There's a compound that we study called PHIP. That's the short name for it. It's a heterocyclic amine that's produced when you cook meat at high temperatures. So any kind of meat, red meats, chicken, doesn't matter. And it's a carcinogen. So studies have shown, animal studies, some human population studies, that people that eat a lot of meat that's well done, that's cooked at high temperatures, have a higher risk of breast cancer, colon cancer, and also we study prostate cancer. And one of the things that's been interesting to us is we hadn't really thought of until recently how much more complex it is than just people that eat well-done meats and this thing they think gets metabolized by the liver, that it's carcinogen and causes cancer. But it actually turns out that there's microbes in the GI tract that metabolize this chemical on their own. And we don't know much about it. There's certain species, one of which is called Intercoccus, that might be related to certain diets. So, some people on certain diets that have certain microbes that eat meat that's been cooked at high temperatures might be at higher risk than others. So it's very complex, but it's just something that we're just now starting to realize.
Rob, you work on this at the very sort of deep level. Can you bring it together a little bit more for us?
Sure. So what's interesting about what the microbiome tells us about diet is that it explains why it's so personal to each of us. So despite the billions of dollars that have been spent looking for clues to why we respond differently to different diets in the human genome, we're all 99.99% the same, whereas your microbiome and mine might be 90% different in terms of the 100 most common species there. And so a lot of what you're seeing at the moment is explanations for things like the idea that you should eat more vegetables, you should eat less red meat, that kind of thing. But your grandparents also knew those things. And so we can't really claim this as new findings based on the microbiome, right? We can claim them as new explanations for why it works. But what's coming, and what's very close, like we can do it in the lab already, is telling people at very fine-grained levels what particular plant they should eat, or that kind of for their microbes. And so there's companies like Day 2, for disclosure, I'm on the scientific advisory board, but they'll already take a sample of your stool and sequence that and then give you a very detailed report at the level of should you eat sweet potatoes or regular potatoes for your blood glucose? Should you eat rice or ice cream? Because it turns out that some people do better on ice cream than rice, at least.
So how do you grow more of the ice cream eating?
Yeah, well,
but
that's what we don't
just
know.
though, Rob, that is very important if you're diabetic, but that's just looking at glycemic control.
Correct.
So there's certainly other aspects to health, even in diabetics, beyond what your blood sugar is.
Absolutely, and the reason why this exists for glycemic control is you can have a continuous glucose monitor and get readings every minute, and get a very detailed account of how each individual food is changing your physiology and your microbiology. So the problem is that for something like inflammation, or even worse for something like weight loss, there's the same variation one person to another. And at an epidemiological level, we can connect it to the microbiome, but it's much harder to do those detailed studies of individual food items at the individual level. It just takes a lot longer if your readout isn't every minute.
David, your place, WashU, and specifically Dr. Gordon's lab there, have done some really fascinating work looking at the sort of deep sequencing, combining it with machine learning to reach some incredible conclusions. Can you fill us in a little bit on what some of that work looks like?
It's the progress in this field has been incredibly impressive. And I think at this point in time, I would start out by saying that I really think we can now imagine taking a pill that repopulates our intestine with a certain set of bacteria that will promote leanness. Or we can imagine a pill that has the substrates for the bacteria that are already there that would promote leanness. Or I think we can even imagine, and Rob sort of got into it a little bit, taking a sample of each of us and using that sample with certain tools to determine the right diet for the particular health issue that we may have, whether that's malnutrition, which is of course a massive public health problem, or obesity, which is another major health problem, or even begin to imagine beyond those diseases, some of the complex diseases that you've heard about. So in the Gordon lab, many labs have made this progress. I'm going to say something about some of the recent findings from the Gordon lab, which is at WashU and has been a leader in this area. In particular, what the Gordon lab championed was the use of germ-free animals, germ-free mice, germ-free pigs, which can be repopulated with bacteria in a controlled fashion to do experiments. And secondly, deep sequencing and machine learning algorithms to analyze the data, because you're looking at trillions of bacteria and you're looking at differences in the food that is going in that are massive variables. So, and the Gordon lab published a number of years ago that you can transfer the microbiota from twins who diverge in obesity or leanness and reproduce that in a germ-free mouse. So you can take the microbiota from somebody who's obese and turn the mouse's metabolism into the obese metabolism.
The illustration for that is quite
similarly, you
dramatic.
can do that. So this was the proof in principle of the germ-free animal as a place to test what I call preclinical testing. And then this lab published four observations this year that are, in my opinion, mind-blowing observations. First, they used a machine learning algorithm to identify 15 very rare bacterial strains among trillions that correlated with recovery from acute malnutrition. That signal was then used to do a clinical trial in Bangladeshi infants that were malnourished. And the trial actually compared the usual treatment of acute malnutrition with a treatment that was concocted in a germ-free mouse. So the treatment was concocted in the germ-free mouse by populating the germ-free mouse with samples from the malnourished infants and then testing certain foods to see which would be the foods that would generate the good 15 strains, complement of 15 strains. And what was discovered in this case was, and the foods that were tested were culturally acceptable to the Bangladeshi community. And the results showed that the certain things that were used ordinarily in Bangladesh to treat children with diarrhea and malnutrition were very bad for their recovery. But a set of foods, of 5 foods, was determined and shown to, over a 30-day period, improve the recovery of the infants. So
these
idea, these were called by Jeff and his group microbiome defined complementary diets.
And what were some of those foods? Give us some examples.
Well, in this particular case, the most effective included chickpeas, banana, soy flour, and peanut flour. So those foods represented the ideal concoction for recovery from acute malnutrition. Now, this is amazingly deep science, quantitative science, that had to be used to do this.
Wow.
They recently published 2 other papers that are mind-blowing, and I'm sure Rob knows about these. In one paper, they analyzed different fiber preparations to see which fiber preparations, using the germ-free animal paradigm to test it, which fiber preparations would promote more of Bacteroides strains. And this goes back to their original study in which they showed that Bacteroides strains played a key role in the obese metabolism. And, in the lean metabolism.
From in the obese.
In the lean metabolism. And when you say lean metabolism, you mean the metabolic byproducts of the bacteria and then the change in the host's physiology. So in this study, they showed that there were components of what particular fiber preparations led to, which could promote the Bacteroides strains to become dominant and to lead to the lean phenotype. Implications of this are that certain fibers can actually be identified that promote the lean, and of course, as we know, that's a big problem for us.
David, I'm going to stop you there for a moment and ask Rob to comment on some of this research.
Yeah, well, if I could comment briefly, I agree that this is absolutely groundbreaking work, being able to link particular items from food to particular bacteria that you want to promote and then have such a profound impact on child health. I want to make sure that you know that it's not just metabolic traits that you can transplant by transplanting the microbiome. So with Sarcus masmanian at Caltech, we've been able to show the same sort of transplant from humans into germ-free mice of analogues of multiple sclerosis and Parkinson's disease and autism. And other people have been able to do things like rheumatoid arthritis, major depressive disorder. So what's really compelling about that is there are many, many different kinds of traits that you can transfer from a human to a mouse by transferring the microbiome to establish causality. The other thing is that in both mice and humans, we can radically transform those microbiomes with diet. In mice, it just takes one day. In humans, it usually takes a few months. But the prospect for changing your microbiome into a good configuration with the right diet for you is shaping up to be very compelling.
And I want to just emphasize Rob's point about diet, because even though this sounds incredibly optimistic and exciting, you can take a pill and become lean. Or as a friend of mine called me up the other day and said, I want you to gather up some microbes from a skinny 18-year-old and bring them down to my house and give them to me. I said, I wish I could do that, but we're not quite there. But I just want to emphasize the importance of diet in maintaining all of this in terms of colonization and significant repopulation, so that it's not just a matter of transferring the microbes, and we see that with fecal transplant, with FMT all the time. The diet is really essential.
Yeah. Absolutely, otherwise it's just like sprinkling the seeds on a parking lot and they're not going to grow there, right? You need soil, you need to fertilize them.
One of the other experiments that was done this year, which is fascinating, is utilizing this understanding, which comes from really sophisticated scientific analysis, to determine what in whey protein actually leads to the kind of bacteria that would degrade some toxic product of processed foods. This actually leads to the proof of the idea that we could, by manipulating the microbiome, either by prebiotic, probiotic, or by diet, it writ largely into preventing a side effect from a drug that you would be taking.
And David, do you want to talk about your other colleague Ted Steppenbach's work using diet and the microbiome to prevent flu?
The study, the recent study showing the byproduct of bacterial metabolism.
Yeah, correct. Where the one where you have the plant flavonoids, and then if you have the right bacteria, it degrades them into a compound desaminotyrosine, and that reduces. So then normally if you infect mice with the flu virus, you kill 95% of them. But if you give them this compound as well, half of them live. So you massively increase survival if you have the right diet and the right microbes.
Yeah, and it's just, there's been enormous hype about about this. And what we're seeing with high quality science is that the hype is reality here. That's what we're saying.
Karen, I'd like to ask you, you work in the prostate cancer area. Are we at the point where we can take a man with prostate cancer and we can reverse engineer back to figure out how to mitigate those factors that cause the cancer to develop? Is that where we are?
Unfortunately, the short answer to that is no. The only accepted risk factors for prostate cancer are advanced age, family history, and being of African American race. And so those are none of those things are things you can change, right?
What about insulin resistance? Isn't that?
Well, right, so there are other risk factors that have been looked at that aren't defined quite yet. That would be one of them. Also things like obesity, smoking. In terms of, if you could take a step back in time and say, what could you do maybe to prevent prostate cancer? One of the things that we study in the lab is inflammation in the prostate as a risk factor for prostate cancer development. And that's a very broad term, I know, but we know in other types of cancer that inflammation is a bad thing, that it serves as a risk factor. It can definitely cause cancer. And I think maybe one of the curiosities with prostate cancer has been that there's a lot of different things that can cause inflammation in the prostate. When we study infections in the lab, microbes that come from a microbiome that we didn't even know existed 10 years ago, and that's a microbiome that exists in the urinary tract. If you've ever heard a doctor or anybody really say that urine is sterile, has anybody ever heard that? It's absolutely not.
More sterile than my neck of the woods with stool for sure, but,
right.
but definitely
urine's not sterile.
Yes.
And there are microbes that reside in the urinary tract. And in terms of prostate cancer, the urinary tract or the urethra runs through the prostate. So we think that the prostate can be exposed to anything that's running through the urethra. So we study infections from the urinary microbiota as a risk factor, but then also, there are certain dietary components that can cause inflammation. Smoking that can, you wouldn't think of smoking as causing inflammation in your prostate, but it can. So I guess the short answer to your question is no, there's not one thing I can pinpoint. There's no wear your sunscreen to prevent prostate cancer, unfortunately. But I think.
In a Cleveland Clinic, we a clinical trial that looks at both PSA and also gene expression patterns in prostate cancer patients who are in the watchful waiting or observational periods. And we found that 2/3 of those patients using anti-inflammatory whole foods diet with omega-3s, vitamin D, that there's a 2/3 reductions in PSA and reversal of all the gene expressions that have to do with cancer.
You reminded me that my favorite example of the foods in prostate cancer is the geographic variability in prostate cancer incidence. So in the United States, the incidence of prostate cancer is very, very high. A lot of men, unfortunately, will develop prostate cancer. In Asian countries, not so much. It's very, very low in Asian countries. But when Asian men come to the United States and start eating our terrible Western diet, their incidence of prostate cancer skyrockets. So there's something there. We don't understand it yet, but there's got to be something there. It could be environmental, but we think it's...
There's a great study of Polish women who, from Poland, eat 30 pounds of sauerkraut a year, which is basically a probiotic food.
The most potent probiotic.
And they have very low rates of breast cancer. When they move to the United States and they stop eating it, they actually get much higher rates of breast cancer.
We see the same thing with inflammatory bowel disease, with Crohn's disease and ulcerative colitis. And if we look at India and Southeast Asia, we see very low rates. And when those populations move to the US and adopt the standard American diet, the rates go up. Speaking of India and Southeast Asia, Rob, you had a slide illustrating this incredible increase in autoimmune diseases, sort of explosion of modern plagues. Can you talk us through that a little bit and this whole hygiene hypothesis idea?
Yeah, absolutely. So I'm sure you're all familiar with Rachel Carsten's work in the 1960s, the idea of silent spring, where attempts to combat individual species of insects with DDT had all these unintended consequences for the ecosystems, where especially birds were unintentionally wiped out. So there was no bird song in the spring. And what we're concerned about in the microbiome is that we're seeing the same thing going on in our inner ecosystem right now. And my colleague Marty Blazer, who's now at Rutgers, has this wonderful book called Missing Microbes that goes through all the ways that we're depleting our inner ecosystems through antibiotics, through cutting ourselves off from microbial exposures in the natural world and so on, through increased use of C-section, all of these different things that deplete our microbiomes. Low fiber diets is another one.
Antibiotics.
I think I said antibiotics, but if I missed it, I'm sorry.
Proton pump inhibitors.
Right. And then there's all kinds of drugs that no one knew affected the microbiome until the last 3 or 4 years.
SSRIs turned out to be a big group for everyone on an antidepressant. Might want to reconsider
And
that.
acid blockers, yeah.
Yeah, atypical antipsychotics, all kinds of things that you'd never suspect were impacting your microbiome. So what's fascinating is over the 20th century, as one infection after another was brought under control by advances in public health, in medicine, and so forth. At the same time, there's this explosion of chronic diseases. So things like type 2 diabetes, inflammatory bowel disease, cardiovascular disease, multiple sclerosis, I think, is on that graph. And what's fascinating is that when the graph I used in the slide was published back in 2002 in the New England Journal of Medicine, none of those diseases were known to be linked to the microbiome. But today we know that all 4 of them and dozens of others can not only be associated with the microbiome in humans, but you can also transplant them from humans into mice using the techniques that David told you about with respect to obesity and malnutrition. So we can establish causality, at least in the mouse model.
Yeah.
And we're now doing things like trying to predict not just what your state looks like today with the microbiome, but what it's going to look like in the future. So we've been working with the FINRISK cohort, where they have stool samples from 2002. And what we're doing is we're taking those 2002 samples and trying to predict every bad thing that happened to that population medically for the next 17 years. And so we can get pretty good associations with things like all-cause mortality from the 2002 sample. So not just reading out your current state, but saying what's going to happen going forward if it's business as usual. And what we need to be able to do is apply the same sort of technology that Jeff Gordon has been able to apply in malnutrition at the start of life. We need to be able to apply that throughout the lifespan to make sure that you're getting the food and the microbes that you need to keep you healthy throughout the lifespan. And if I could just comment briefly, so when we look at populations like the Hadza, hunter-gatherers, and the Yanomami, they have completely different microbiomes from anyone who's taken up farming, let alone anyone who lives in an industrial society like this one. And things like the Verruco microbia, the Hadza and the Yanomami and other pre-industrial groups don't have them at all. So it's really interesting that they seem to be healthy in our context because they're not something that we've significantly co-evolved with as a major player in our gut. And so understanding what we need to farm and what we need to rewild in our microbiome, that's really compelling at the moment.
Yeah.
But even in our Western civilization, there's still studies that show rurality, exposure to farm animals, for example, is preventive, can distinctly reduce the incidence, for example, of a child developing Crohn's disease. And we know that antibiotic use is a major risk factor for the development of autoimmune disease, particularly antibiotic use early in life. And that perfect storm of being born via C-section, where you don't have the benefit of all the population through the vaginal canal, not being nursed, where you don't have the benefit of important ingredients in breast milk that further feed the baby's burgeoning microbiome, and then recurrent use of antibiotics early in life. This is a perfect storm for developing a lot of these diseases. There's a genetic predisposition, but really environment is the trigger for the gun.
Yeah, and that's another more hopeful way to frame that, though. So if you have a C-section and you have early life antibiotics, if you can then breastfeed, that erases the effect of those early life events.
Yeah.
So it's very modifiable.
Yeah.
But then the problem is being able to confer those benefits for families who are unable to breastfeed.
Yeah, I mean, breastfeeding is interesting because you look at babies who are breastfed versus bottle fed. The breastfed babies have high levels of something called butyrate, which is one of the most important nutrient fuels for the gut. And there are starches in breast milk that are non-digestible by humans and that are used for the microbiome. When you breastfeed, you get the butyrate. When you don't, you get high propionate, which is actually a neurotoxic short-chain fatty acid that's been linked to autism. So, I'm a clinician primarily, and for the last 25 plus years, my focus has been functional medicine, treating patients by focusing on the gut. Even before we knew a lot of this data, it was really clear to me that many of my patients, whether they had symptoms or not, that the best way to help them with most inflammatory diseases, whether it was autoimmune diseases, asthma, digestive disorders, neurodegenerative disorders, mood disorders, autism, ADD, was to focus on the gut. And by doing a very simple protocol, which is sort of, optimizing the diet, removing all the bad stuff, putting in some pre and probiotics and some nutrients to help fuel the gut, we had extraordinary results even before we sort of knew as much as we do now.
What we were doing.
Yeah.
Mark and I had this conversation before when I was a medical student at Columbia. I remember being in my pediatrics rotation and seeing a sign up on the wall saying breastfeeding prevents Crohn's disease. And I remember thinking, that's those La Leche people again who are at. This is crazy. How can breastfeeding prevent Crohn's disease? That's absurd. Well, again, it turns out this ingredient that Mark is talking about, the third most populous ingredient in breast milk, human milk oligosaccharides, is there to increase the production of butyrate. So it really comes full circle, and widespread education about this stuff, and of course alternatives for women who can't breastfeed.
Yeah.
Now, Robynne, you mentioned farm animals before, and I just want to come back to that because that's really important, not just for Crohn's disease, but also for asthma and allergies. And there's a great story that again was in the New England Journal a couple of years ago by Erika von Mutius in Munich and Jack Gilbert, who's now at UC San Diego. So the Amish have very low levels of asthma and allergies, but then the Hutterites, who are another religious group that come from the same region of Germany and are very closely related genetically, have very high rates. And it turns out that the difference between them is the Amish, the young children go out on the farm, whereas the Hutterites, there's a very strict division between farm life and home life, and only the men are allowed to go out on the farm, and they wash themselves very thoroughly before they come indoors. And it seems like, and then looking at all the sources of possible microbes, including the microbes in the dust in the homes, it turns out that what's important is not the chickens and not the dirt from the crops and so on, but specifically the microbes from cows and from hay that cows have been eating. Yeah, and we're doing a study with Erika in Bavaria at the moment, where you can go down a row of houses, and one house will have a barn where they bring the cow in the winter, and then the next house won't. And at that level of resolution going down the street, the families that have cows that come into their house have much lower rates of asthma than the families without cows. So clearly the recommendation is if you want to protect your kids, you should get a cow. And that's not so practical, right? And so then the question is, could we put the cow in a bottle, or could we have could we have a cow the size of a hamster, or a hamster that produces the right antigens or whatever?
But it does seem like so much of what the nut we're trying to crack is how to go back to the cave without going back to the cave. So how do we have a more natural relationship with the outdoors? There's a great book called Nature Deficit Disorder, Last Child in the Woods. How do we get our kids away from the screens and out into dirt and into the cowshed, ideally, to prevent these autoimmune diseases? And I think there's this incredible balance, because industry oversells it, in my estimation. Take this pill and you'll be fine. And the medical community and scientific community is sometimes a little bit slow to translate these findings into actual clinical guidelines. So how do we balance it? David, as the distinguished dean and vice chancellor of medical affairs at WashU, how do we create more balance between this sort of industry overselling and medical and scientific slow to adapt?
Here I was saying that the hype was realized. I do want to caution about some of the specific disease associations, because I don't want to give the impression that we believe that the microbiome causes the disease. I think it's more we believe the microbiome plays a role, often in many of the diseases that we have a hard time with now. They're very complicated, and that's the reason why we have a hard time with them. So the microbiome plays some role, and many of the studies are association studies that show that. The big thing we have to do now is stick with the hard science. This is a very complicated area. There are trillions and trillions of bacteria they each feed off of each other and they feed off of the host, and there is all kinds of crosstalk between them. We also know there are viruses and fungi in the environment of the microbiome, and those we are only just beginning to understand. It's a little harder to work on the viruses and the fungi than on the bacteria, and so that's trailing behind a little bit. They're very interesting suggestions about the role that specific viruses play in the population, and they may play them through their interactions with bacteria. So complicated system needs really hard science, but I believe we have the tools now, and that's sort of what I was describing. We have the tools now to do the right experiments, and those include the ability to characterize the bacteria using sequencing, our ability to use big data approaches to analyze that, and then very careful experiments that can lead us in the right direction. The work that's really exciting, though, is this work that has to do with general overall health. And that, I believe we really are, we really do have the science that tells us how we can manipulate the diet, how we can manipulate the microbiome for those more general phenomenon, the metabolism that is associated with leanness, the metabolism that is associated with obesity, and we know those two things play an outsized role in disease. So I think the answer to the question you asked me, stick with the hard science. We need more of the hard science using preclinical models to get at specific therapies.
When we were in a panel together earlier this year, you talked about what you can do to modify, just to bring it back again to some of the hard science, to modify those patients who have a poor response to immunotherapy, the prostate cancer, and it really just kind of stopped me in my tracks. It was so fascinating. So I'd love you to just share that with the audience for a minute.
Well, I mean, I think in terms of just to take it a step back, you know, I mentioned it earlier, I think that mechanistically we still don't understand how exactly the gut microbiome is mediating response to this immunotherapy, which is trying to kill a cancer that's not in the gut, that's someplace else in the body. I don't think we really understand what's happening there. We know that certain species of bacteria, if they're present or if they're not, might seem to influence whether or not you respond. We just don't know how. But I think some things to keep in mind are that the GI tract is actually one of, if not the largest immune organ in your body. There are so many immune cells. You know, you think of your like the spleen or your thyroid or thymus. But there's a lot of immune cells that exist in the GI tract, and the microbes that are there are constantly being sampled by the immune system. And not only that, the things that microbes produce are constantly being exposed to the immune system. So a lot of our overall, general immunity, general health takes place right there in that interaction in the gut. And everything that we eat is being seen by all of those microbes there, and they're metabolizing it to all sorts of things. We're only barely touching the surface of understanding what's going on there. So I think in the absence of knowing what specifically is mediating that interaction, one of the strategies that is now being tried in clinical trials, and I'm involved in one that's taking place in Oregon, is to do fecal microbial transplant, FMT, from patients that have responded to immunotherapy into patients that have not responded, to see if in the non-responder you can now boost their response to immunotherapy. And I know that this is being done right now in melanoma. I've heard, from the PIs that are running these trials that so far things are looking good. It waits to be seen, and our trial in prostate cancer is just open.
I see. So you don't have results back yet for prostate cancer?
We just started there.
Yeah, I think what you said is really critical for people to understand. So 60% of your immune system is in your gut, and there's one cell between you and a sewer, and that's held together with these little things called tight junctions, which are easily disrupted and separated. So the food and all the bacteria supposed to go through the cells, but it can go in between them and get directly exposed to the immune system. And all the diseases that everybody's talking about are inflammatory diseases, whether it's Alzheimer's or autism or depression or autoimmune disease, obesity, diabetes, heart disease. These are all diseases of inflammation, and that's where the linkage is, I think. And I think we know that many of the things we do in our life, whether it's being exposed to glyphosate, which disturbs the microbiome, or environmental toxins, our low fiber, high sugar diet, the antibiotics, C-sections, lack of breastfeeding, NSAIDs, PPI drugs, steroids, hormones, all these things disrupt that. And it's easy to get a disruption in that barrier, and we call that, increased intestinal permeability, or the generic term is sort of leaky gut. And that's really a central focus in functional medicine, which is how do you clinically address this, even if we don't understand everything, if we understand the basic principles of how do you grow an inner garden, how do you create a healthy gut, using common sense, simple things that are low risk, we can have profound clinical impact. So while the science, I think, needs to progress and will, and we're literally at the infancy, I think, of the field, I think clinically there still can be a lot done with patients to help them recover from many of these chronic problems by focusing on gut health.
I really concur with Mark as a clinician. I've gone in the sort of 22, 23 years of my career from treating primarily the sorts of things you can feel and see and touch and put your finger on, like gallstones and colon cancer, to now this very sort of microscopic array of conditions of leaky gut and small intestinal bacterial overgrowth, et cetera. But primarily, most of our treatment is based on, number one, judicious use of pharmaceuticals, so really peeling back the 20 supplements people are taking that they don't need, and the constant onslaught through things like acid-suppressing drugs and antibiotics and SSRIs, et cetera, and then also diet. And it is incredible the amount you can accomplish with those two things, with really getting rid of all the potential things that disrupt that intestinal permeability. And
we know things that
diet.
help repair it, and we know the things that impede it. And so clinically, there's a lot you can do with nutrients and foods and various fibers and probiotics. So even though I think that I completely agree, we have to advance the research, we also have to realize that we know enough now to start to help people clinically help with some of these chronic conditions. That's what I do, and I think I can share case after case, but it's really striking when people come to see me, the first thing I do is focus on their gut. I take a very detailed gut history. I want to know all the things and exposures from when they were born to what their mother ate, to all the way through, and any insults. And you can often connect the dots by doing that, and you can often then start to reverse engineer what happened and then prospectively help them rebuild their gut and rebuild their health.
Now, Robyn, I want to come back to something you said that I thought was very interesting, where you said that when you were starting out, most of the conditions you were treating were things that you could see and touch, whereas now it's all stuff that's invisible. And in some ways, you can think about that as being a problem of user interface, right? It's a lot more difficult to grapple with the conditions that we have now. And everything you heard from David and Karen about needing really solid, really technical science is true. But if you think about, say, the cell phone that many of you are playing with at the moment, it's based on this incredibly deep technical knowledge. It's based on a tremendous amount of radio engineering, satellite engineering, polymer chemistry, all of that kind of thing. And it's covered by literally 500,000 patents. But you don't have to care about any of that stuff to take a photo of your kid and send it across the world. And so I think if we're really going to realize the potential of the microbiome to benefit the lives of billions of people, the way that collection of technologies in your cell phone has already benefited the lives of billions of people, we need to pay a lot of attention to the user interface. We need to figure out how to make all the stuff visible. We need to figure out how to give you feedback on when you're going in the right direction or the wrong direction. And all of that takes a lot of artificial intelligence and machine learning. It also takes a lot of human factors design to try to make all of that information accessible to clinicians and ideally to individuals. I don't want to say patients, because ideally you want it to be the sort of thing that could apply to everyone, even when you're not sick.
It's true. At lunch, Mike Milken was talking about these Japanese toilets that actually take stool samples and can real-time measure your microbiome. So you can actually, like a Dexcom glucose monitor, measure the response of your poop to what you're eating every day.
Yeah.
Or you could just eat more kale every day. You could do that
You could
too.
what?
Well,
You could just eat more kale every day.
There are also the new approaches with nanoparticles that you can ingest. And will actually will actually give you a readout of particular metabolites. I'm sure that'll be one of the strategies to.
That's the other extraordinary thing, Rob and I were talking about. We know we have all these metabolites, and there's 37 billion reactions every second in our body. And our genes make proteins, and all the chemical reactions are a result of that. But our microbiome has 100 times as many genes as we do, and they're also producing metabolites, which we're absorbing. And if you took a blood sample, he said we estimate that 30% of the samples of metabolites in your blood come from the microbiome, the metabolome of the microbiome, and it may even be more, you said.
Yeah, what's really amazing is if we compare germ-free mice to mice that have their regular microbes, throughout their whole body, you see all these metabolites that are produced or altered by the microbiome. And even in the brain, something like 2/3 of the unique kinds of chemicals that we see are different between the mice that have microbes and the ones that lack them.
Yeah. So you're not you, you're really you're a host for your bugs.
You're really an amalgam.
We're just
of
like
both.
basically 1% human.
Right, a big tube to feed them.
But the bidirectionality is also fascinating because our microbes are affecting us and we're also affecting our microbes. There have been, Rob, I'm sure you can speak more to this, the experiment where they were able to induce anxiety in mice by giving an inoculum of Campylobacter jejuni. And I think about all the post-infectious irritable bowel syndrome I see in my office where people say, not only are my bowels different, but I feel different. I'm more anxious and I'm sort of different psychologically than I was before this infection.
Yeah, that was a fascinating study, although you don't have to do it with a pathogen. So we did some work a few years ago with Chris Lowry with a different bacterium, Mycobacterium vaccae that comes from soil. And what we saw is when you give that to mice and then you expose mice to social stress, either by putting them in a cage with a big mouse that beats them up, which they don't like very much, or even if they just watch that through a glass barrier, they don't like it either. And they stop eating, their hair falls out, they won't associate it with other mice. But if you expose them to this particular kind of bacteria from the soil before you apply the social stress, they're very resilient and they bounce right back from it. And that's linked to that inoculation, preventing the bad changes to their microbiome that normally happen with social stress.
Yeah.
So what I think is really interesting about that, this idea that gardening is good for your blood pressure and good for your health, it's a very old idea. And some of it's got to be the satisfaction of seeing the plants grow and being able to eat the fresh fruits and vegetables. But increasingly, we're thinking that some of it might be that direct contact with the soil.
The forest bathing studies from Japan really substantiate that. They took two groups of workers. One group they had on their lunch break walk around in an urban area, and the other group they took to the forest to walk around through the trees. And they found tangible, measurable improvements in things like blood pressure, not just subjective things like feelings of well-being, but blood pressure, heart rate variability, et cetera, showing a significant decrease in stress and anxiety in the forest bathing group. So get dirty and do it outside.
Yeah, and doing it outside is really important. So, so in the nucleotides to neighborhood study, which is one of the things that we ran by our American gut at UC San Diego, what we found was that just the number of minutes you spend outdoors has a significant influence on how diverse your gut microbiome is, which is fascinating, right? Because it's a place where canonically the sun does not shine, and yet somehow being outside affects its diversity.
Yeah, it's amazing. Yeah, I think that's so true.
The mind-body, the gut-brain connection is also really well illustrated with the example of toxo. So Toxoplasmosis gondii is a really common parasite. I think probably about 30-35% of people in this room have it. In France, it's as high as 70%. And most of us, when we acquire toxo, we don't have any symptoms at all. We might have one day of loose stool or so, but we're usually asymptomatic. In mice, there's a slide I like to show of a mouse kissing a cat. And I say, why is this mouse kissing a cat? Cats are the natural enemy of mice because they prey on mice. The mouse is infected with toxo. And when mice get infected with toxo, the toxoplasmosis essentially hijacks neurons, brain cells, in the amygdala of the brain and cause the mouse to lose their fear of cats. Not only do they lose their fear of cats, they're usually repelled by the scent of cat urine. The mice actually go in search of cats. Why? So they can get eaten, because toxo cannot reproduce in mice, only in cats. And to make it even more sort of sci-fi, there have been studies now showing that people who have obsessive compulsive disorder and some people with schizophrenia are infested really, or sort of infected with a very, very heavy level, heavy inoculum of toxo. And so there's an association now that's developing between obsessive compulsive disorders and toxoplasmosis infection. So as Mark said, most of these conditions are inflammatory, and I would argue that a lot of them are also infectious too. So think about things like anxiety and depression, and where does this stuff come from? And heretofore we felt, well, it comes from our parents, right? It's all their fault and it's genetic. But we know, again, the genes are the gun, the environment is a trigger, and how many of these things, liver cancer, hepatocellular carcinoma, that's an infectious condition. So I would, put it to Karen, is prostate cancer ultimately going to turn out to be an infectious / inflammatory disease?
I mean, we think that it is. We're working on the proof of that, but you're asking a very highly biased person that question. Also with liver cancer, though, even though hepatitis obviously is a risk factor for liver cancer, there's also interesting work being done showing that high-fat diet and basically something called the translocation of parts of different types of the not so good bacteria that are very inflammatory, very immunogenic. It goes into the bloodstream and it gets into the liver and causes inflammation there as well, which promotes liver carcinogenesis. So again, there's a link, even in an example where that there's also a viral relationship there, there's still, this relationship with what's going on in the gut and high-fat diet promoting the expansion of the wrong types of bacteria that can be very pro-inflammatory.
But it also depends on the fat, right? If it's omega-3, it doesn't. If it's trans fat or other fats, polyunsaturated fats, it does.
David, I see you're smiling. I'm coming right to you. So one of your areas of expertise is aflatoxins and liver cancer. And there's a strong dietary component there, if I understand it right, too, with the exposure to aflatoxins and people eating a high-meat diet and so on.
Peanuts.
And peanuts, of course.
Yeah, I mean, there are different mechanisms for liver cancer.
Yeah.
So there are viral-mediated mechanisms. There's even genetic diseases in which proteins accumulate in the liver will predispose to liver cancer. So I'm not sure I would say that the microbiome is going to be causing all those different types of cancer.
It's a little bit of overselling, I would agree with you.
Yeah.
Yes. Although the study was the children, I believe it was in the Philippines, who were exposed to the moldy peanuts, who developed liver cancer. The poor kids did not develop as much liver cancer, and it turned out because the more affluent kids were eating a diet higher in animal protein and animal fat, and that was potentiating the effect of the aflatoxin. Maybe, like the TMAO data, maybe microbially mediated, but certainly not for sure.
So when we think about causality, it's really interesting to remember that to the Victorians, tuberculosis, as they called it, consumption, was a classic example of a genetic disease that was inherited. And the reason why is pretty simple, right? So back then, everyone in London was exposed to Mycobacterium tuberculosis. So if you came down with the disease or not, it was about your host genetics. Whereas now we think of it as completely an environmental disease because most people aren't exposed to the pathogen. So the variation in whether you get TB or not is going to be explained by exposure, not by genetics. And other things that they also had huge genetic research programs in were goiter and cretinism, which also ran in families, and rickets, which also ran in families. Because if everyone's iodine deficient or everyone's vitamin D deficient, then the variation you see is going to be in your genetic susceptibility to those diseases. And so it would have been tragic if those research programs had continued and it was mostly about telling people that they were genetically inferior if they had TB or if they had rickets, rather than understanding that the way to address those particular diseases was with antibiotics for TB or with nutritional supplements for rickets or goiter. And I think there could be, I think there are going to be lots of cases like this where microbes are one piece of the puzzle. They're not always going to be the piece that you can use to transform the disease, but sometimes they are. And I think a lot of the cases where they are, we just don't know yet.
David, I'd like to, in the remaining minutes, touch on an area that I think lots of people are interested in, which is intermittent fasting, and how intermittent fasting might interact with the microbiome for disease prevention, for weight loss, et cetera. I know one of your areas, again, is autophagy, and autophagy, sort of loosely translated as eating oneself, which I think of as sort of a third cousin of intermittent fasting. And is there a connection there on that spectrum between intermittent fasting, autophagy,
well,
disease
I don't
prevention?
know. Rob may know. What we're referring to when we talk about autophagy is the vacuum cleaner of the cell. So this is a process by which we clean our cells every day. All the bad proteins, the bad lipids, the parts of the inner parts of our cells, which are called organelles, which are worn down by use. The system also is responsible for removing invading microbes. It's in every cell. And what we now know from very, very powerful research is it declines with aging. And so many age-dependent degenerative diseases are a result of cellular poor health because this system is not operating. And what caused you to associate that with caloric restriction, intermittent fasting, is that we know the autophagy system is activated by starvation, caloric restriction. It's also activated by exercise. And there's a lot of data that suggests that the positive effects of caloric restriction and of exercise on longevity and aging are mediated by this pathway. So much of my research is on drugs that activate this pathway as potentially slowing age-dependent degeneration. The relationship to the microbiome, I don't know of very much work in that area.
No, I think it's
And
not well known.
is it possible to imagine that there are metabolic effects of the microbiome that factor into the autophagic response? Absolutely.
Well, there's clearly a link between the microbiome and mitochondria, which is where a lot of the autophagy sort of...
I would disagree, Mark. There's a mitochondrial autophagy, and then there's an autophagy of everything else in the cell.
Yeah.
So a lot of people have focused on mitochondrial autophagy in Parkinson's, for example, but that's only one of many aspects that autophagy hits on.
So very briefly, Sachin Panda has shown good links between intermittent fasting and the microbiome, but whether that's linked to autophagy, I don't know.
So in the literally final seconds, I'd like to ask each of our panelists to give their 20-second or less formula for good health. Mine, based on what you've told me, is get outside, into the cowshed, get sweaty, and eat more vegetables. Mark?
I think it's, understand that you have an inner garden and learn how to take care of it and grow a good garden through all the things we've talked about.
Rob?
I agree with Robynne's generic advice, but personalized advice is coming, although it's not there yet, despite what companies are going to tell you today.
Exercise and diet. Exercise and diet.
Karen?
And I would say it's not going to be solved by taking some pill, exercise and diet.
And maybe a fecal transplant.
Or maybe it's just a thing. And then exercise and diet.